Armando Hasuringan, Biology and Medicine videos. Please make sure to subscribe, join the forum and group for the latest videos. Please visit Facebook, Armando Hasuringan. Please like, and here you can also ask questions, answer questions, and post interesting things, including your artworks. Please send me your artworks. You can also change the quality settings to the highest one for better graphics. In this video, we will talk about antigen recognition by phagocytes and B and T cells in a lot more detail, a bit more detail than the previous videos. We begin by starting off with a progenitor lymphoid cell that can give rise to either naive B cells or naive T cells. Here, this progenitor lymphoid cell will give rise to two naive T cells. These two naive T cells are CD4 and CD8 naive T cells. And also this progenitor lymphoid cell will give rise to a naive B cell. These naive T cells have T cell receptors on them that can recognize antigen. If a T cell recognizes a self-antigen, this naive T cell will be destroyed before it matures, because we don't want a T cell to attack our own body, because self-antigen is our own body. Now let's follow this other naive T cell. When the naive T cells are developed from a progenitor lymphoid cell, they all have CD4 and CD8 co-receptors. However, when they mature, when they proliferate and they differentiate, they will either become a naive CD4 T cell with a T cell receptor or a naive CD8 T cell with a T cell receptor. The only difference is that a naive CD8 T cell has a CD8 co-receptor here, and a naive CD4 T cell has a CD4 co-receptor here. So let us follow first what happens with the naive CD8 T cell. Well, the naive CD8 T cell will become activated when an infected cell, infected antigen presenting cell, such as a phagocytes, present an antigen on an MHC class 1. So the phagocyte will present a foreign antigen to the naive CD8 T cell via the MHC class 1, which stands for Major Histocompatibility Complex class 1. This will activate the naive CD8 T cell to become a cytotoxic T cell, or a CD8 cytotoxic T cell. Cytotoxic T cell, they destroy infected cell with the same specific antigen as was presented by the infected phagocyte. So CD8 T cells kill infected cells. That is their goal when they're activated. So what about naive CD4 T cells? Well, a naive CD4 T cell will become activated when a non-infected antigen presenting cell, such as this phagocyte, presents a foreign antigen on an MHC class 2, a major histocompatibility complex class 2. This naive CD4 T cell only recognizes a specific foreign antigen. And when it's okay, it will become activated through this MHC class 2. And the naive CD4 will become activated to become a T helper cell. A T helper cell, its main goal is to enhance the immune response by activating B cells as well as activating natural killer cells and also macrophages or other phagocytes. So that was how naive CD4 T cells and naive CD8 T cells become activated through recognition of specific antigen presented to them. So how about the naive B cells? Well, naive B cells have antibodies when they become naive B cells from the progenitor lymphoid cell. The naive B cells' antibodies can recognize specific antigens of a pathogen. So, for example, this naive B cell will recognize this antigen of this specific bacteria, and then this will cause the naive B cell to engulf the pathogen and then process it, and then present this foreign antigen on an MHC class 2 to the activated T helper cell. So the B cell engulfs this foreign pathogen, processes it, and then presents its antigen on the major histocompatibility complex class 2 to the T helper cell. The activated T helper cell can then activate the B cell to proliferate and to differentiate into either memory B cell or a plasma cell. Plasma cells secrete specific antibodies towards that antigen. Memory B cells form memory of that specific antigen so that a next infection by the same antigen can be resolved quickly. So what do these antibodies do that are secreted by the plasma cells, for example? Well, these specific antibodies that recognize these specific antigens can do three main things. First of all, they can neutralize a pathogen. So for example, if here was a healthy cell and here was a bacteria that is about to kill it, antibodies can bind onto the antigen of this pathogen and essentially neutralize it. So it prevents bacterial adhesion. Secondly, these specific antibodies can opsonize a pathogen. So for example, this bacteria, through the antibodies can opsonize it, basically coats it for phagocytes to easily engulf it. So opsonization essentially promotes phagocytosis, such as macrophage phagocytosis. Thirdly, specific antibodies towards a specific antigen can activate complement. Activating complement essentially means that antibodies can opsonize the pathogen, promoting phagocytosis, as well as lysing a pathogen. So antibodies activating complement can opsonize a pathogen and also cause the pathogen to lyse, to self-destruct, to burst. And that was the antibodies, the function of antibodies secreted by plasma cells. What I just drew just then was just an overall picture of how the CD4 and CD8 T cells become activated and how the naive B cell also become activated. Now, let's go into it in a bit more detail. And we can answer this question: how do the B and T cells recognize only those specific antigens? Because there's so many different types of antigens with different amino acid sequences. How is this possible? To understand this, we have to look at each of these individual cells, the naive CD8, naive CD4, and the B cells in a bit more detail individually. Let's begin by looking at the naive B cell. The naive B cells have antibodies on their surface, on their cell membrane. And these naive B cells have specific antibodies that only binds to a specific type of antigen. So what gives the antibody such diversity to bind to a unique type of antigen or different antigens? Well, let's zoom into this antibody here and have a look. This is an antibody. The antibody consists of a variable region on the N terminal of the antibody and a constant region on the C terminal. The constant region determines what class of antibody this is. So it can be a IgM, IgD, IgE, IgA. The variable region can be composed of many different types of amino acid sequences that gives it its uniqueness. So antibodies can be all different and all unique when they are made. So now, let's see how it works. Let's just take this antibody of this pathogen and look at it at an amino acid level. Here is the antigen, the same antigen of the pathogen. Now, the antibody recognizes only specific sequences of the antigen. This specific sequence is known as epitope. So an epitope are sites within the antigen that the antibody recognizes. And through this recognition, the B cells can then engulf it, or the B cells can then destroy it with the antibody, whichever one it is. Now let's look at the naive CD8 T cell. The naive CD8 T cell, remember, becomes activated when a foreign antigen is presented on a major histocompatibility complex class 1 by an infected antigen presenting cell. such as this phagocyte. So the naive CD8 T cell has a T cell receptor. The T cell receptor also has a variable region and also has a constant region, just like the antibody. The variable region also is made up of different amino acid sequences that can bind to a specific type of antigen. Okay, so this naive CD8 T cell becomes activated when a infected cell presents a foreign antigen on the MHC class 1. How does this come about? Well, if we have an antigen presenting cell, such as this phagocyte, and it is infected by a pathogen, so here we have a pathogen infecting this phagocyte, the pathogen will start accumulating inside the cell and basically taking over the cell. However, during this time, the infected phagocyte will actually also process this pathogen and then take its antigen. So process its antigen and then present it on a major histocompatibility complex class 1 on the cell surface. So this major histocompatibility class 1, MHC1, with this unique antigen will then present it to a naive CD8 T cell. And this naive CD8 T cell has to recognize this specific antigen with its T cell receptor and also its CD8 co-receptor. And if it's all okay, this naive CD8 T cell will become activated to be a cytotoxic T cell. And so this naive cytotoxic T cell will then kill cells infected with that specific antigen. So that was naive CD8 T cell. How about a naive CD4 T cell? Well, similarly, the naive CD4 T cell has a T cell receptor that has a constant region and a variable region. And so the variable region can be composed of different types of amino acid sequences that therefore will bind to different types of antigens. And remember, we have different types of, we have many CD4 T cells, so the variety is tremendous. So the naive CD4 T cell gets activated when a non-infected phagocyte, antigen-presenting cell, presents a foreign antigen on an MHC class 2. How does this happen? Well, we have a antigen-presenting cell here, such as this phagocyte, and it will engulf this pathogen through phagocytosis. So the pathogen doesn't actually infect this phagocyte, but the phagocyte will engulf it, will phagocytize it. And when it phagocytizes it, the pathogen will be essentially inside a vesicle and be destroyed by lysosomes, through acidic content. And so the pathogen will lyse and then will basically form bits of antigens. And through this, the antigen-presenting cell, this phagocyte, will process these antigens and then present it on a major histocompatibility complex class 2. And then it will present this major histocompatibility complex class 2 with the antigen to the naive CD4 T cell. The naive CD4 T cell will only recognize a specific type of antigen through its T cell receptor. So luckily, also in this case, this naive CD4 T cell's T cell receptor recognizes this specific type of antigen. And also during this process, the CD4 co-receptor has to confirm it. And then from this, the naive CD4 T cell will become activated to become T helper cell, which will then activate the B cells, etc., etc. And that concludes the video on how the naive CD4, naive CD8, and naive B cell becomes activated, and how the antigen-presenting cells, such as the phagocytes, activates these lymphocytes. I hope you can appreciate and understand the immune system from this video. I'll provide links as to how the B cells, naive B cells, get its diversity, the antibody diversity, and as well as how the MHC class 1 and MHC class 2 gets processed. And also other links I'll provide in the description box that you can click to learn more about the immune system. Thank you.